Stem cells.
Stem cells are undifferentiated cells, meaning that they don't have a specific job. Their purpose is to replace other differentiated cells that have been harmed or are no longer functional. There are several types of stem cells. There is embryonic stem cell, which is a natural stem cell. That may sound kind of confusing, so let us explain. Embryonic stem cells are found in the embryo and are totipotent, meaning pretty much all of their genes are active, so they have the ability to turn into one of the 220 different cell types in the human body. As the embryo develops into the blastocyst, the stem cells lose some of their ability to turn into any cell, so they become pluripotent.
Then there are adult or somatic stem cells, and these contain different categories. There is the iPS cell, also known as the induced pluripotent stem cell. These are adult stem cells that have been reprogrammed to have the abilities of an embryonic stem cell. They need this ability because somatic stem cells have less active genes. In previous years, they were reprogrammed by retroviral transduction, which is a genetic transduction which is defined as being a transfer of non-viral genetic material within a viral code. However, that has fallen out of favor, and the favorite now is non-viral, non-integrating, plasmid-based reprogramming systems. iPS cells came out in 2006, but now there are CIPS cells, which reprograms the cell's function only using chemicals, which lessens the chance of mutations. Also, they express the SAL4 gene earlier than the iPS cells. SAL4 is needed for limb regeneration.
Finally, there are SAP stem cells. SAP stands for Stimulus-Triggered Acquisition of Pluripotency, and the creation of these cells does not involve genetic manipulation. Rather, it involves stressing them, meaning dipping them into slightly acidic solutions or squeezing them through a tube of cells. But what makes these stem cells special is that they can give rise to placenta. A stem cell's DNA is less compact than a differentiated cell's DNA. Scientists found that there are 4 genes active in a stem cell that are not active in differentiated cells.
Moreover, when these genes are reintroduced into a cell, it can turn them back into a stem cell. How stem cells differentiate into other cells is different. In regular cells, the inactive genes always remain inactive and cannot become active during cell division. However, stem cells have high potency, meaning how many active sites they contain. Therefore, since they have a high potency, they have the ability to be many kinds of cells, and as they start to differentiate, they lose their potency, or become limited in their ability to be a different cell type.
The process of a stem cell becoming a differentiated cell takes progeny, and transcription factors are essential to how the stem cell will differentiate because they have to go through asymmetric cell division so that one daughter cell can have the ability to still remain a stem cell and the other begins to differentiate. The factors that cause stem cell differentiation in vitro are chemical and physical. To name some, there are growth factors, cell culture substrates, co-culture environments, three-dimensional culture, and signal inhibition. Scientists aren't really sure what activates the differentiation of adult stem cells in the body, though it's usually because of injury.
In the pea-glow lab, we genetically transferred the green fluorescent protein, also known as GFP, into the E. coli bacteria through plasmids. Only when the bacteria containing GFP was in the plate with the arabinose, ampicillin, and lauriabrot did the bacteria glow. The presence of the arabinose caused the repressor gene to change shape, so that it could no longer fit on the operon, allowing RNA polymerase to transcribe it. Stem cells relate to this lab because, like the bacteria in the plate with arabinose, under the right conditions, a stem cell will be able to differentiate into any specialized cell. Another reason stem cells are related to this lab is because some scientists use viruses to insert certain genes into the stem cell DNA, which is similar to what the plasmids in the pea-glow lab do.
What we would like you to take away from this video is that though there are several ethical issues, stem cells are important for future research and can provide the cure to many fatal illnesses. One of the ethical issues is that embryonic stem cells kill potential life, but many women lose most of their eggs anyways. Another is that it's not natural, but in a way it is, because scientists can use the stem cells from your own body to cure you. And this would help with diseases because your medicine could be individualized instead of the generalized way.
I hope you guys learned a lot about stem cells. We thank you guys for watching, and this video was brought to you by Anna Zenas and Ashley Lyaprin in the Biology Honors 418 class.